Muy buenos días a todos, bienvenidos a esta segunda mañana de este gran Congreso Nacional y Simposio Internacional 2021 de Sorgo. Bienvenidos a todos. Les recuerdo, tuvimos un martes con todo, con muchísima información hablando de Sorgo en dos módulos espectaculares con muchos técnicos, muchos especialistas hablándonos y te vuelvo a recordar que queda todo editado como para que lo vuelvas a ver. Hoy es miércoles 24 de agosto, año 2021, y vuelvo a repetirlo porque al quedar todo editado queda para la posteridad, ¿no? Queda para que otros lo sigan viendo, lo sigan recordando, sigan informándose de muchas cosas y de todo lo que se vino diciendo con lo último en tecnología, en mejoramiento, en usos, en manejo de este gran cultivo. Bien. Voy a presentar el módulo 3. El módulo 3 tiene como auspiciante a la empresa Advanta Semillas, a quienes le agradecemos, por supuesto, por este auspicio y por su presencia acá en el simposio y congreso. Es un módulo dedicado a tecnología y mejoramiento genético. Ayer estuvimos viendo mucho el contexto del cultivo, vimos sus usos. Bueno, ahora nos metemos de lleno en la tecnología y en el mejoramiento genético, algo que ha crecido enormemente, yo diría, en los últimos 4 o 5 años a través de este gran boom de la siembra y el uso de tecnología en sorgo. El coordinador de este módulo 3 es el ingeniero agrónomo Santiago Rentería, que es egresado de la UBA, especialista en mejoramiento genético y senior breeder de Advanta Semillas. Santiago, el módulo, todo tuyo.
Muy buenos días, bienvenidos al tercer módulo del Simposio Nacional de Sorgo. En esta oportunidad vamos a tocar el tema de mejoramiento genético vegetal y tecnología. El primer disertante que tenemos en el módulo es William Rooney. Actualmente es miembro de la cátedra Barlow Monsanto de Fitomejoramiento y Mejoramiento de Cultivos, profesor y miembro senior de la Facultad AgriLife, Departamento de Suelos y Ciencia de los Cultivos, de la Universidad de Texas, A&M, por sus siglas en inglés, poniendo especial énfasis en su investigación en el cultivo, en el mejoramiento del cultivo de sorgo y en su genética. El doctor William Rooney, a lo largo de su amplia trayectoria, ha contribuido a desarrollar y formar académicamente a más de 25 Magister Science y más de 30 doctorados, y tiene más de 200 publicaciones en trabajos científicos, revistas internacionales, y autor de patentes actualmente vigentes en el cultivo de sorgo, y ha contribuido también al desarrollo y liberación de germoplasma de amplia utilización en los programas de mejoramiento. Su charla va a ser adopción de tecnología para mejorar el proceso de mejoramiento en sorgo. Lo escuchamos.
Okay, thank you very much for inviting me to speak to your meeting today. It's an honor to be able to speak at this meeting. I've heard about this meeting for many years, but not had the pleasure to do that. And even though it's in a distance, it's a pleasure to be able to talk to you today. We're going to talk about a couple of different topics. A lot of the interest in our program right now is on looking at refining the breeding process of sorghum. And so I've chosen 3 research areas that we have been active in the past few years regarding modifications or impacts to the breeding process and what we produce in sorghum. And I want to cover those briefly for you today. Specifically, we're going to talk a little bit about chemical gameticides. We're going to talk some about gene flow to Johnson grass, and then we're going to do a very short introduction into some work on genomic selection that we've been partaking of here in the past few years. So my goal is to give you some insight into what we're thinking about when we talk about how does a sorghum breeding program what does it look like in a few years as we see adoption of new technologies to speed the process, make it more efficient, and make it more effective? So this slide pictures my apologies to the authors of this manuscript. In the rush to get this presentation done, I forgot to put credits to them, but this is showing haploid induction in corn, the process used to do that. primarily what we see done here, if I have a pointer, is a field of corn. You'll note that tassels are removed from a large proportion of this, and an inducer line is placed in here to induce haploid formation, whereby in corn they can use color markers to discriminate between the haploid progeny that are induced versus the diploids. This is obviously, y'all are familiar with the idea that this speeds the breeding process and makes things more efficient, and that we definitely like to do something like this in sorghum. In fact, projects have been funded and modest successes in the development of these technologies have occurred in the past few years. At some point, haploid induction and developed doubled haploids may be a viable part of a sorghum breeding program. And this led us, when we started thinking about this, into the concept of, well, how are you going to make the seed on the F1 cross to be able to identify the haploids? And that came from that, we began to realize that we've got a problem that corn doesn't have. Corn detassels to emasculate. In sorghum, we don't have that luxury. And in this case, we cannot use traditional male sterility systems because that would result in a haploid that was male sterile as well. So we needed a temporal method of chemical fertility control. And that's where we got into the search for a chemical gameticide. And I'm here to report to you today on one that we found that works quite well in sorghum. It's called trifluoromethanesulfonamide, TFMSA, originally described in maize by the Dr. Lossert from the Pioneer Group at the time. And we have tested this as one of several that we looked at as candidates and found that this was the most effective in sorghum. And that work has been published in an earlier phase a few years back. Mode of action, we don't know a lot about it, but the suspected interference of transport of proline to the anther is what causes a normal B line to be completely sterile. Here's the B line version of this G line, as we call it, compared to an A line, resulting in very high levels of, if not complete sterility. And so this technique we've used now in our program for the past 3 years has been quite effective in inducing sterility that is temporal. We wanted to know what the effects of this temporal stability might be on products that are produced using that. And so I refer you for full details to a paper we recently published in Crop Science by Imani Kianam, who is a graduate student in our program, in which she actually compared the effects of TFMSA on plants, the AB parents themselves, as well as hybrids derived from that. And you see the specs here on what we did, but she evaluated 23 different AB pairs in which we were comparing the cytoplasmic male sterile version of these lines with a chemically induced sterile version of the B line, which we designate as a G line here. And you see the dosages. And what we're interested in is what's the effect on the inbred line, and then what's the effect on hybrids produced therefrom. And so you'll see that we produced a set of hybrids in this study that we evaluated in the 3 environments, and we compared hybrids made with cytoplasmic male sterility with those made with TFMSA. Our expectation that the mode of sterilization would not affect the productivity of the resultant hybrids and their progeny. And we hope that the null hypothesis as the same would be manifested in this case. And so we evaluated them over a period of time. In looking at the effects of this work, we did notice that there was an effect of TFMSA on these particular inbred lines in terms of productivity. You see here under controlled pollinations with bags, we see a reduction in the G line relative to the A line in grams of seed produced per panicle. We see that trend as well in open pollination. We see a slight delay in anthesis of about 3 days, and we see a modest reduction in height of about 10 centimeters. So these things do this chemical does affect the growth of this plant, delaying maturity, reducing height, and reducing seed yield overall. But what you'll note is that it is as effective in terms of inducing sterility as that cytoplasmic male sterility system is. So we have good seed sterility at some maturity, height, and a little bit of seed yield cost. If we look at the hybrid performance, what we see there is basically no difference between these two sets of hybrids. This is across the all sets of hybrids. And if you look at basically any parameter except plant height, you see no statistical differences. For plant height, we saw a significant statistical difference in that the G lines were 2 centimeters shorter on average than the A-line hybrids or the cytoplasmic male sterility hybrids, which is really not enough to be of any consequential effect in a relative breeding program. Interesting self-progeny. We saw no difference in terms of the frequency of self-progeny, which were monitored in this particular study by having white-seeded females cross to red-seeded pollinators and then counting straight numbers of panicles that were white-seeded in the resultant hybrid seed. And you'll see that both of those were below 1%. If you look at 3 locations just for yield, then you see the same trends, that these were consistently not different between the 3. So we can safely say that the G lines and the A lines perform comparable, and the G line hybrids are just as good for comparison purposes as A line hybrids. We saw similar results when we looked at individual inbred lines and how they ranked in that yield trial. And you see that for the most part, if they were ranked in the top 5 with the cytoplasmic male sterility, we saw very consistent results there, with 4 of the 5 being there, although there are some rank shifts in those, but not a major amount of change. And you can see from these pictures that there's really not a lot of difference in some of these particular hybrids. If you look at the hybrid on the top, that's the cytoplasmic version. On the bottom is the G line or TFMSA version of that hybrid, looking very similar. So we view this as an opportunity to start evaluating material and having some significant applications to this in breeding and test crossing new seed parents before sterilization and the investment in that process. We've used it for hybridization and genomic selection experiments already. We've used it for breeding crosses, and we've even used it for some wide hybridization work that I'm going to talk about here in a little bit. So there are many different applications of this from a breeding program. And there are many different methods of applying this. We've done it in the greenhouse, we've done it with a hand sprayer in the field, and we've done it with tractor applications where we've done whole field sections. Timing of that application, these are detailed in the publications that I've mentioned already. And there are some considerations there. But overall, this has been very successful for us in our breeding program, and it's now an integral part of our breeding program. I do want to say a little bit about safety on this. Here's the safety data sheet on this particular compound. You will see that it is something that we want to treat as a pesticide. It's not the most toxic things out there, but it is something that we have to be considered as hazardous, just like any pesticide that we might use in our programs. And so we handle these just like we would handle pesticides with similar hazard identification, using PPE and managing applications as appropriate for that. I say that, and I will conclude with this section saying that we think that TFMSA is effective to induce that temporal male sterility, and we are using it from an experimental standpoint. And I put that in here because we have not, and nor do we have any intention to move forward with an official label for this compound. We use it on research projects and in research projects. Doesn't mean someone can't, and anyone in here is free to do so if they see such applications from that standpoint. Switch a little bit now to a second topic of gene flow. Gene flow has become important in sorghum, and we're talking about gene flow from sorghum to Johnson grass. primarily because we see now some traits coming into sorghum and commercialized, notably herbicide resistance, as you are all familiar with, that is now moving into the commercial setting. And there is legitimate concern about maintenance and the maintaining the identity of these products for both protection and to avoid them moving into these wild relatives where the presence of those could be problematic in production systems by providing resistance to this particular species, Johnson grass in particular. And so there's other traits obviously under development. And so the question is, will they move to Johnson grass? How will they move to Johnson grass? And should we be concerned if they move to Johnson grass? And you can look at this from several different situations, because that gene flow and that interspecific hybridization can occur in a field where they both present, and they could occur in both directions. Sorghum is the female, Johnson grass is the female. They could occur after harvest with volunteers in that field. They could even occur in feral sorghum growing on roadsides. These last two are particularly problematic from a sorghum as a female standpoint, because remember, typically these volunteers that occur on the roadside or in the fields after harvest are F2 progeny from a hybrid that is made with cytoplasmic male sterility. If you'll remember, then a fair number, about 25% of all of these progeny that are growing on the sides of the roads are then going to be male sterile. And if Johnson grass is around there, they provide a natural crossing block for this potential gene flow with sorghum as the female. So we joined with Dr. Muthu Bhagavathian from our weed science program in Texas A&M over the past few years to look at these gene flow rates with sorghum as a female and with Johnson grass as a female. And I'm going to briefly report to you on some work that Cynthia C.S. did in her master's study with Dr. Muthu and myself on these different rates. And so the first thing we did is sorghum by Johnson grass, in which we put out blocks of male sterile sorghum and the same A-line male sterile sorghum, as well as B-line fertile sorghum in an endemically infested a field endemically infested with Johnson grass. And we allowed these to cross-pollinate or self-fertilize in the case of the bee lines, and allow cross-pollinating, asking, are there differential rates of interspecific hybridization due to the sorghum genotype? And so we did this for 2 years, generating seed from these A-line blocks and B-line blocks, and then evaluating them for the frequency of that material in the seed that was produced from that. And here's a close-up of those blocks. On the left is a male sterile sorghum with Johnson grass pollinating it. On the right, a fertile sorghum with Johnson grass to pollinate that. And the results were revealing. Over the 2 years of seed production, here you see the list of 12 different sorghum A-B pairs that we evaluated, all from the Texas A&M sorghum program. And you look at the outcrossing frequency, and this outcrossing frequency is the frequency of interspecific hybridization with Johnson grass. So of the seed that is produced, how much of it is actually a sorghum by Johnson grass hybrid? And by genotype, you see very stark differences between these different seed parents. Some of them are essentially zero, while others are as high as in 2019 with 623, a high of a little above 1.2% of the seed produced on that plant was of a Johnson grass hybrid. Alternatively, in 2018, 626, a relative of 623, was quite high. Others that trended similarly, but we did see that year by genotype interaction. The point being is that both the year and the genotype conditioned these 1% to 1.5% maximums of outcrossing in a male sterile sorghum line. Now understand, we did not have sorghum pollen as competition in here. These were 100% male sterile. And so most of these were designed to look at what's the maximum rate of interspecific hybridization we would see. Alternatively, in the B lines, they performed consistently over both years. And so you see one note here, one graph with some of the same lines showing up as higher frequency than other genotypes. Now, you need to note that the y-axis on this is an order of magnitude lower than that we see with the A line. So crossing when you have fertility is much, much lower, an order of magnitude less, but nevertheless, we see these similar trends. Certain genotypes outcross at higher rates than other genotypes. Now, why is this? Well, when you start to look at the progeny that are produced and what types of progeny are produced, this is a split on that. And so you see in the first column that the 12 genotypes on the left, the number of progeny evaluated for that genotype under flow cytometry to look at the ploidy of the progeny that were produced. And I want you to look at these two right here because these are the interspecific hybrids. You'll note we produced some haploids as well, which are not interspecific hybrids. Those are simply haploids of sorghum. So this is another way to produce haploid progeny in some situations. The vast majority, 95% of all hybrids produced, actually higher than that, 97% if you just look at those and take the haploids out, are tetraploid. Only 3% were triploid. We see the same trends, a little less dramatic here with 76 and 24 between tetraploid and triploid. Now remember, if we cross Johnson grass with sorghum, we should be looking at, with sorghum as the female, a haploid gamete coming from sorghum and a 2N gamete or a 20 chromosome gamete coming from Johnson grass. That would give us a triploid. That's what we would expect, but that's not what we see. We see a preponderance of tetraploids. The reason for this, we hypothesize, is 2N gamete formation in the sorghum gamete. And when you have 2N gamete formation and you pollinate with Johnson grass, it selects those and advances those because these triploids are failing in the development of the endosperm and they don't survive. And so there's what's called a triploid block in interspecific hybridization, and this is clear manifestation of that. This has implications in terms of fitness of these progeny that are produced because these have a balanced chromosome number. And in case when we look at some of these progeny, they are actually fertile and not sterile as most triploids are. So this has some fitness effects, and you see a clear difference in the frequency. Remember, 626 had large numbers and almost all of them. So we hypothesize that 626 produces more 2N gametes than something like TXRH1 or TX642, which had essentially no outcrossing to Johnson grass. The point being here, different seed parents can impact the rate of gene flow to Johnson grass. Some are more conducive to it, others are less conducive to it. Now we're going to flip the system and talk about Johnson grass as the female parent, sorghum as the pollinator parent. To do this, we had a real challenge because we had to have a way to screen these to identify them. And this was always a challenge because if any of you have looked at progeny from this cross in this direction, it is very difficult to confirm that you have a true hybrid. They look like Johnson grass. And so this Ensign sorghum pollinator was a key to this because it allowed us to screen Johnson grass seed, and any hybrids that survived had resistance based on that herbicide resistance trait being passed from parent to progeny. We've done this 2 seasons now, and what we're seeing is definitive confirmation that you have gene flow in this direction. Sorghum can pollinate Johnson grass, And you get anywhere, the first year we had about 1% gene flow or interspecific hybridization rate. And this is with the fertile Johnson grass, and 0.5% frequency in the second year that we did this work. Like we just saw with the females, we looked at those progeny to see what their ploidy level was. And exactly like the female, sorghum is the female, we see that the progeny are tetraploid in a vast majority. In fact, all of these were tetraploid. We found no triploids at all in this situation. So what we're seeing is the same trend, that likely 2N gametes is the key to this. Now, what we have not done is screened enough different genotypes to know if there's variation among these male pollinators for this rate of outcrossing. We are doing that study this summer to follow up on that. What's interesting about all of this from impact is looking at why it's so difficult to find the cross interspecific hybrids of halopints by bicolor, with halopints as the female, compared to bicolor by halopints, with bicolor as the female. And this picture summarizes it perfectly. If you look at the hybrids on the outside of this picture, you see a phenotype that most of you would say, well, that's not sorghum per se, and it's definitely not Johnson grass per se. But if you look at these progeny in the middle here, they look much more like Johnson grass. And that's what we see when we use Johnson grass as the female. The hybrids are essentially indiscernible from Johnson grass, whereas the ones with sorghum as the female parent show a range of variation and clear impact of sorghum on that phenotype, with wider leaves and thicker stalks and a little less retooning in some cases. And the panicles look like an intermediary hybrid between them. I say that because this is going to be a challenge when we have these situations arise. So I'm going to jump forward to my last topic now. I've got a few minutes left, and that's genomic prediction in hybrid grain sorghum. I'm not going to talk a lot about this, but I do want to start the conversation that our group is working in this, and I will refer you to the full details of the two slides I'm going to show you here to some work by Jalis Fonseca, who is a recent PhD graduate program of our program. In the plant genome, just recently published. He had a factorial design with a modest number of hybrids generated from the collaboration of our program with Dr. Ramasamy Parimal at Kansas State. And we set up a factorial to produce these 100 hybrids in groups of combinations of Texas by Texas, Kansas by Kansas, and then the combinations, with a broader goal beyond genomic prediction to look at the interaction of germplasm from different elite breeding programs. That will be a separate paper that we will publish that is accepted for publication, but I'm not going to talk about it today. We grew these, long story short, in 10 environments, 6 in Texas and 4 in Kansas, and used the data around height and yield and maturity to look at building genomic prediction models, which we have since tested in larger field trials and greater sets of environments. And I'm not going to go into great detail on this, except to say that genomic prediction models in sorghum are effective. If you look at the graph on the left, this is the correlation between prediction and the validation, the training set and the validation set based on predictions from a training set, and then comparing it with the actual data that we had from the trial across these 10 environments listed down here at the bottom. And the lines just help you follow this. This red line, solid line, is the correlation between predicted and observed for grain yield across these hybrids across the 10 environments. And you'll note that they range from somewhere in the upper 0.3 to as high as 0.8 across those. And across all environments for the males in these pollinators, depending on which prediction training models we were using, we achieved with the engagement of genotype by environment interactions a correlation of 0.625 for the pollinators and a 0.57 for the seed parents. For And you see those two graphs differing. The blue is the pollinator lines and their predictions ranging from 0.4 to 0.8 as well. So that's why these are both in a similar range right there in the middle. For height, which is a more highly heritable trait, we see better prediction effectiveness with this genotype by environment interaction engaged in the prediction model, giving us the best performance as expected. So we came away from this and have tested it in subsequent additional models that these genomic prediction models are effective, just like they are in corn, in predicting agronomic trait productivity in sorghum hybrids. And that if we include those G by E effects, it improves our prediction accuracy. And finally, a last note that he noted in his, I'm not getting into it at all, is that hybrids adapted to target environments are better at predicting than unadapted hybrids. That's not too surprising if you think about it logically, but it was one of his take-homes that we were able to confirm. For more details on this, I would refer you back to his recent publication and several others that will be following this coming out of our program. The goal in all of this is sort of bring many of these technologies from the logistics of producing haploids through the logistics of genomic prediction in hybrid sorghums and using this for breeding, building breeding crosses, etc., into sorghum programs. I think it's going to revolutionize and change how we fundamentally breed sorghums. It's happened in corn. It's going to happen now in sorghum, and we're right at that point. And when you bring along phenomic prediction methods as well, all of this is poised to see this change in these different methods and approaches we use to breeding the crop. So in conclusion, hit on the highlights, TFMSA is going to be a chemical gameticide that will effectively induce temporal sterility in most sorghum genotypes. We found multiple applications for it in our program. Gene flow is something that will happen. We do know now that we can affect it and we can mitigate it in certain ways by careful choice of parents and identifying those parents that have it happen at lower frequencies, certainly on the female side, and we strongly suspect the same thing will occur on the male side. And we're still looking at the fitness of these hybrids that are produced and how that's going to affect what we see. So I'm cautiously optimistic that we can control gene flow to an extent that we will be able to have traits and see them in a real situation without excessive concern that we're going to lose them quickly right back to the weeds. And then finally, genomic selection, I think, is going to be deployed in the very near future in sorghum. It's going to require a rethinking of how we do things in most sorghum breeding programs, what approaches from both a crossing and a testing that we will be using. And all of these things are just at the forefront of looking at new ways of doing things faster, more efficiently, so that we can get more numbers through the program, so that we can push that game from selection faster by both time and numbers in the program, while maintaining programs with similar budgets to what they currently have. And with that, I'm going to thank you for listening, and I appreciate again the opportunity to participate.
Muchas gracias, William Rooney, por la disertación, muy interesante, los temas, y agradezco tu participación. La segunda charla del módulo de mejoramiento va a estar a cargo del doctor David Jordan. David Jordan es mejorador y genetista de sorgo con una amplia trayectoria, tanto en el sector público como el privado. Actualmente trabaja en la Universidad de Queensland, Australia, como profesor de genética y mejoramiento genético vegetal. Durante los últimos 15 años ha dirigido programas públicos de mejoramiento de sorgo y ha trabajado en la parte de licencias, licenciando alrededor de 2.000 500 líneas, empresas que son o que participan en híbridos, principalmente Australia, y también en algunos programas de mejoramientos internacionales. Se reconoce y ha contribuido de manera significativa a la ganancia en productividad que ha logrado con estas introducciones, y su beneficiario principal son los productores, principalmente en Australia. David Jordan también ha publicado muchos trabajos referentes científicos, tiene alrededor de 130 trabajos en revistas de renombre internacional, en sorgo y una amplia gama de disciplinas. Vamos a escuchar la disertación del doctor David Jordan, que va a hablar sobre mejorando el sorgo para un mundo más cálido y seco. Lo escuchamos.
Hello, my name is David Jordan. I'm a professor in plant breeding and genetics at the University of Queensland in Australia. I'd like to start by thanking the organising committee for giving me the opportunity to talk to you about my work on sorghum breeding. And I'd also like to express my apologies for not being able to be present in person. Argentina is a country that I've always wanted to visit. And at one point in my career, I was employed by a commercial seed company in Australia with a base in Argentina. And we exchanged germplasm and all around the world. And germplasm from Argentina tended to be the most likely to perform well in Australia, and I think vice versa. So I think that's because we have similar problems in some cases. So here's a map of the two production areas in Argentina and Australia. You can see that the Argentinian production area stretches much further south than ours, whereas our production area goes further north. And I think our rainfalls tend to be lower in general. So we have a large geographic area, but a relatively small planted area. And all of our production relies on stored soil moisture. We have highly variable environments, both in terms of weather and soil, and a diversity of management systems in terms of planting time, row spacing, etc. So our planting can occur any time from late August through to February, depending on the location. Mean yields are typically around 3 tonnes per hectare, but vary from less than 1 tonne to greater than 10 tonnes per hectare. I lead the public sorghum improvement program, and this program is funded by sorghum growers, the state government and the federal government. And the objective of this program is to increase the profitability for sorghum farmers and downstream industries by increasing productivity, managing risk, and protecting yield and decreasing costs and increasing crop value. So today I'm going to talk primarily around increasing productivity and managing risk, focused on water and to some extent heat. Because of the highly variable environments, we have quite a different philosophy to improving yield in Australia. It's around the strong focus on the interaction between genotype and management for particular environments. So what we're trying to do in terms of crop improvement is to identify good combinations of varieties and management practices that will work well in a system where the resources that we can spend on searching for these combinations is limited. As I said, there's a lot of environmental variation. It's something that we can't do very much about. So we just need to accept the environments that Mother Nature gives to us. To illustrate this point, I show you some rainfall data from Roma, which is a sorghum growing area. over 100 years of climate data, you can see that there are these periods of wetter than average and drier than average that occur. And in this case, we've got 5 years where rainfall is around, above 700 mils per year. And then the previous 4 years, the rainfall was well below 400 millimetres per year. And a farmer needs to be able to be profitable under both circumstances with effectively the same set of varieties. So one of the ways that farmers tune their systems is through management. We can do agronomic research to identify management systems such as planting dates, fertilisers, spacing, populations that may be suitable for a particular environment. But the value of these systems really depends on the genotypes that are grown in the systems and the types of environments that are experienced by the system. So we can choose management systems and select the best if we know what the best is. Similarly with varieties, there are many different types of varieties that are possible, but the value of a particular variety depends on the management system in which it's grown and the patterns of environments that are experienced by it. We can change these varieties and select the best if we know what the best is. Unfortunately, the challenge about identifying the best combination is the interaction. There are large numbers of combinations of genotype or variety and management and environment. That means that plant breeders and agronomists are forced to work on components of the system in isolation and therefore must ignore many of the interactions. And these interactions turn out to be very important. So where does productivity in cereals come from? Here's a great example that illustrates the point that I'm talking about. This is a study of maize production from the maize genetics from the 1930s to 2000. The dots and triangles represent hybrids that were dominant at the time. So these are hybrids from the 1930s. These are ones from the 1940s. These are the ones from the 1950s. And the different symbols, circles versus triangles, represent different types of management. This is low intensity or management from the 1930s, lower populations, lower fertiliser input. This is more modern management with higher fertiliser and higher populations. And what you can see is that from this baseline from the 1930s, if one was to take the 1930s varieties and grow them in the modern management system, you get a small increase in yield. Similarly, if you grow modern varieties using 1930s management, you get a small increase in yield. The big increase in yield comes from the interaction of modern varieties and modern management. This situation occurs in almost all crops. However, for breeders and agronomists working in Australia, we have the challenge of a much more variable environment. So trying to search for the best combinations is much more difficult. Here we have a lot of environmental variability, both in terms of soil and climate. And so when you plan a trial, the question is, how relevant is that trial to the long range, the long-term average in terms of environments? So how much faith should I put in the results of that particular trial? And also there are too many combinations of traits and management systems that we might want to explore, and that makes it really difficult for us to, identify what's the best and answer questions like, well, what happens if I was to do the same thing but change tillering? Or will I get the same result in another location? So one of the ways that we've tried to deal with this is using computer models, because these can be run at relatively low cost and quickly. So crop models are computer programs that predict the likely outcomes for a crop grown in a particular environment with various combinations of trait and management interventions. And this allows us to integrate the knowledge that we have about variability in climate. So we tend to predict over multiple types of environment, soils and rainfall. We can look at different management interventions. We can look at different genetic interventions, and we can look at the interaction between those two things. So we can understand the types of environments that occur and give our trials some relevance. And we can identify the best bet combinations of genetics and management that we might want to evaluate in real life. So rather than trying all of the combinations, we can focus on the ones that we think are most likely to be useful. So to illustrate this capacity to understand environments, if we look at the top panel here, these are different sets of crop simulations for crops grown in different environments under different management conditions over our cropping area. Each black line represents a crop by management combination. And this y-axis is the supply-demand ratio, and this is to do with water. So it's how much water the crop is able to use relative to how much water it would like to use. So when the line sits at 1, that means the crop is basically getting all of the water that it needs. As it goes down, it means that there's more stress. These graphs are centered on flowering time, and it gives you an idea of where water stress occurs relative to flowering time. And this allows us to look at our different cropping regions and see how frequently different types of environments occur by using climate data for over 100 years. So that allows us to deal with some of the, or at least have an understanding or appreciation of some of the variability. But what you can see by looking at these graphs is that typically Australian sorghum is has either a relatively low level of stress or it has some degree of post-flowering stress. So the other way that we'd like to try and use models is to identify combinations of genetics and management to evaluate. So as physiologists or as plant breeders, we might come up with an understanding of the environments and then look for traits that might give us an advantage in those environments. And as a breeder, I would go and search for genetic diversity in those traits, potentially introgress it into good material and test the performance of those genotypes. And then if things look really good, we might start to deploy those in a breeding program using some sort of high throughput screening method or phenotyping system with drones or screening with molecular markers. So I just wanted to give you a little bit of an explanation of drought in Australia. Most of our soils have a relatively high water holding capacity. And so you can think about it as we talk about it as the bucket, and how much water is available for the crop. So how much storage is available. Now, farmers will store moisture during the fallow period in that bucket for growth in the next crop. And there'll also be in-crop rain, and the relative proportions of those two sources of water will vary. Water from the crop will be used depending on the environment. So the hotter it is, the more water that gets used. And obviously climate variability has an impact on all of those things. And I guess to some extent, management can influence the water that's in the crop, in the soil due to fallow. So the result is that not all the droughts are the same in terms of timing, severity, and duration. However, because we tend to not plant unless we have good stored soil moisture, we don't often see pre-flowering drought stress. Most of our drought stress occurs post-flowering. And Water stress during grain filling or post-flowering grain drought has major effects on sorghum yields. It reduces grain number, it reduces grain size, it causes lodging and senescence. And here's a really good example of what relatively small amounts of water available after flowering can do to grain yields. And you can see these patches in the soil where there's a little bit more water have actually produced quite a good crop, whereas the yield's much lower in the areas where there's slightly less water. So many of our interventions involve shifting water from its use by the plant pre-flowering to post-flowering. So if we think about the situation where the crop has a finite amount of water available for growth and production, so you can think that you might increase population, which actually results in more water being used for growth and potentially less water being available for production. Alternatively, you could grow a crop where the population was very low, which would reduce the amount of water that was used for growth and increase the amount of water that's available for production. Now, this system is going to be potentially a much more stable system, but lower yielding. This system is probably a more unstable system, but with higher yield potential. And what we're really trying to do is get the right balance of water use pre-flowering and water use post-flowering. And many of our interventions try to achieve that. So too much water use for growth, not enough water use for growth, and a balanced water use. So what are our strategies that we use to breed for terminal drought? Either we try to tolerate the drought, and that's a very difficult thing to do, to find genetics that will allow plants to tolerate drought, or we can try to do things that avoid severe post-flowering drought by ensuring that water is available at grain filling. So we can save water pre-flowering for use during grain filling. We can capture extra water to avoid post-flowering drought stress, or we can use improved water use efficiency, which again makes more water available for that post-flowering drought period. So in terms of saving water pre-flowering, we could change canopy size and duration by increasing population, changing row spacing. Both those management options will change the amount of water that gets used pre-flowering and potentially is available post-flowering. We can similarly do things with genetics, either by altering flowering time, so a longer duration crop is more likely to, will use more water during the pre-flowering period, and that may be useful under some circumstances. Or we could do it by tillering, because tillering changes the size of the canopy. So a smaller canopy will use less water during the pre-flowering period and potentially leaving more water for grain filling. And finally, you can change leaf size. So hybrids with thinner leaves are going to use less water than hybrids with wider leaves and bigger leaves. So one of the ways that farmers deal with this on a management sense is to use wide row spacing. So here we see double skip sorghum where we leave these rows bare, and that reduces and combined with high density to reduce tillering, enables water in this inter-row period to be saved, and as the roots go out, it's available for use during grain fill. This type of system is quite conservative. There's a trade-off in that you don't get as much yield in the good years, and I think the crossover point is around 2.5 tonnes per hectare. But it reduces the risk, but also reduces the potential yield of the system and the potential output in really good years. tillering is another way of change, as I indicated, is another way of changing crop water use. So here we model, this is modeling data at a particular location. The x-axis represents, the yield outcome from the normal crop grown in that year, and the dots or the squares represents the performance of a simulated low tillering genotype. And again, this is very similar to the skip row system in that low tillering is advantageous at lower yield potential, but at the high yield potential, there's a negative impact. So you have this trade-off between productivity and risk, and the farmer with this sort of knowledge can make some decisions. One could also increase population with a lower tillering variety and potentially reduce some of that downside. There's also an interaction. So in those skip row systems, sorghum responds to the low density by increasing tillering, and that tends to make the canopy size bigger and reduces the value of the system. So under those circumstances, a low tillering genotype could actually be much better. One of the nice things for seed companies about low tillering is it enables you to increase the flexibility of the locations that you could potentially sell that variety. So maybe the low tillering variety can be sold across a bigger range of environments than would otherwise be the case. However, low tillering takes away the option of, increases the risk when establishment is poor because you don't get as much compensation. So there are always advantages and disadvantages. So if we now think about capturing more water, we could also do that by management. For example, fallow management. If we do minimum tillage and stubble retention, we get more water infiltration, we get less evaporation, and so the amount of water stored for the crop pre-flowering, so there's less runoff, or pre-planting is increased. Another way of managing that is very common in Australia is this thing called controlled traffic, where all of the mechanical equipment travels down the same path. It actually saves a lot of fuel as well, but it means that the compaction is centered around this area where the tractor travels, and that means that the water infiltration over the rest of the paddock is actually much higher. So compaction is limited to the wheel tracks. We could also capture more water using genetics. So by changing root architecture, we can allow the plant to access more water at depth. And here's our attempt to model the impact of root traits. What we found that in many Australian cropping systems, there is still water available deep in the soil that's not used by the crop. And changes in this nodal root angle are likely to influence water capture in different environments by modifying how much water is captured to the side of the crop and how much water is captured below the crop. Here, we're looking at the modelled consequences of narrow root angle at this location using 100 years of weather data. So in this case, as I said before, this is the yield of the normal hybrid over those 100 years of data. And this is the yield of the predicted narrow root angle hybrid. And you can see that there's a general benefit to this trait across a range of yield levels. Obviously, the impact is lower in the highest yielding years, but there's not a negative impact of the trait, it would appear. In these years, these would be years where there's a lot of rainfall, whereas here you've got some level of stress. So that's an interesting trait to select for, and one that's, at least at this location, is unlikely to have negative effects. So From a breeding point of view, what we did was searched for a range, searched to see how much variation there was, and tried to work out a way to quickly select for root angle. So it turns out that the angle that you measure at the 5 or 6 leaf stage is very predictive of the root angle at the mature stage. We developed these phenotyping systems based on these small perspex plates where we could measure the root angle of large numbers of genotypes very efficiently, and then we've screened thousands of lines, and then we could potentially use that in selection. So here is some data from a diverse set of germplasm, and interestingly, some of the really elite lines from our program have high narrow root angle, and this narrow root angle means they're more likely to, the roots are more likely to go to lower depth. And the range of root angles in the commercial hybrids varies from about 25 to 32, but in material that we test, there are lines that have angles down as low as 15 degrees, and others that have very wide root angles. So there's a lot of opportunity to exploit this trait. And just to demonstrate the impact, these are very large root chambers where we've got a single plant grown about here. And this is the comparison between a wide root angle plant and a narrow root angle plant. It's not startlingly obvious, but the wide root angle plant actually extracts water further away from the where the plant's growing. So it goes horizontally a lot further, whereas the narrow root angle plant actually goes deeper and uses more water at depth. So if you've got a deep soil, narrow root angles are good. If you've got a shallow soil, then perhaps wide root angles could be useful. So, theoretically and from a modelling point of view, it looks like root angle would be an interesting trait. We genetically located regions of the genome that are associated with the root angle trait using data from our multi-environment trials, and this is one of our trials grown at that Dalby location that I mentioned earlier. And what we found was that narrow root angle was associated with this stay-green drought tolerance trait that we select for, and that higher yield occurred in water-limited environments where that trait was present on deeper soils. So the information from the modelling helped us to search for something which actually turned out to be useful. This is a trait, though, that's likely to interact with management and environment. So, as I said earlier, the narrow angles allow the plant to extract water at depth, whereas the wide angles allow you to extract water beside the plant. So you can see that the wide root angle could be useful in these skip row systems, whereas the narrow root angle is much more important in the deeper soils. Okay, now using water more efficiently, there are a range of management things you can do to do this. Make sure that the plants are growing optimally, that they have the right fertilizer, etc., that weed control is done appropriately to avoid weeds using water, and that evaporation is reduced, perhaps by keeping stubble over the soil. At the genetic level, it's possible to improve water use efficiency by changing transpiration efficiency, which is a trait that's related to the way the plant uses water and produces biomass. And transpiration efficiency is really defined as the amount of biomass that's produced per unit of water transpired, and it is a trait of interest to plant breeders that are trying to improve yield under water limited environments. And it can be achieved in a number of different ways. You can change the biochemistry of photosynthesis, and things like carbon isotope discrimination might be a way of getting at that, but it tends to be expensive and hard to measure. You can look at the way the plant responds to environment. So under very hot and dry conditions, the plant transpires much more for the same amount of biomass that it creates through photosynthesis than on a more humid or a cooler day. So if you could make it so that the plant didn't use as much water during that hot, dry period, then you could actually improve water use efficiency. And finally, you can make changes to leaf morphology that will potentially have impacts on the efficiency of the system. So we've looked at modelling transpiration efficiency using different types of mechanisms. Again, this is the same environment. And depending on the mechanism, this trait has a large impact on yield, and usually not too negative at the very high yields, but potentially it has some negative effects in some circumstances. But it's certainly a trait that we should be very interested in. And one of the reasons we haven't pursued it to a great extent is it's difficult to measure. So we set up systems to enable us to measure transpiration efficiency on a large number of plants with the aim of using molecular markers to predict the likely transpiration efficiency of particular genotypes to enable it to be used by plant breeders. And again, one of the nice things about this study is we found that many of the elite lines from our program already had quite high transpiration efficiency compared to genotypes grown or from other parts of the world. So this gave us confidence that this was a useful trait and one that we should continue to select for. Now, going to something different here. So probably not such an issue in Argentina, but in Australia, high temperatures around flowering time can be a real problem. So sorghum is tolerant to high temperatures except around flowering time. And I'll go into the details of why soon, but it's to do with high temperatures causing poor production of pollen. So can we deal with this by management? When we do the simulation, we see that changing planting time to avoid high temperatures is really not an option for us. And so we're really forced to be looking at selection for resistance. So biologically, what's happening is that high temperatures around flowering cause pollen production to be interfered with, and those darker pollens are fertile and the light-coloured ones are basically sterile. And temperatures around or above 38 degrees around this time can cause quite severe sterility in sensitive varieties. And we're actually seeing crop failures due to high temperatures. And this is just a graph showing the relationship between pollen percentage, how much viable pollen is produced, and seed set. So here's an example where two varieties were exposed to high temperatures. This one has reasonable seed set, and you can see there's still some damage. This variety has almost no seed set. And there's quite a range in the variation, or at least the resilience of different genotypes to high temperatures. And so again, this is something that we looked at building screening methods for, and we have a collaboration with with Advanta around that at the moment. But one of the issues around high temperatures is the impact of climate change. Now, I indicated to you that there have been crop failures due to high temperatures. When I started as a sorghum breeder about 30 years ago, we did not see high temperatures causing crop failures very often. It was a rare thing, but it's becoming increasingly common. And some work that my colleagues did looked at, well, what's the likely impact of current and future climate scenarios on the problems that we will see associated with temperature. So there's a range of sites across the sorghum growing area, and these are the range of yield impacts for different periods of time. So the green is 2030, the orange is 2050, and the blue is 2070. And this is particularly looking at a heat-susceptible cultivar, and you can see that there are a range of circumstances now where high temperatures are going to cause yield reductions now, but that there is a general increase in the frequency of high temperature events and the likelihood that those events will cause yield depression, to such an extent that in the future, we're going to see a different profile in the types of environments that are important to us. So while drought by itself remains a problem, it will decrease in severity. And one of the reasons it decreases in severity is that there's an advantageous effect from CO2. However, it will still remain a problem. We will see drought with no heat. We will still have some good environments, but they will be less common. We will see an increasing number of environments where there's heat impacts and no drought. And we will see a lot of environments where we get both drought and heat. So this is really a trait that we need to breed for pretty urgently in the future, or for the future. So I guess the take-home messages, and I know I covered a lot of ground here, but I think the most important one, and probably the one that is relevant across a range of different places, is that the interactions between genetics and management are important and offer substantial opportunities for productivity improvement above and beyond what an individual agronomist or plant breeder might do. Breeding is expensive, and if you can find a management solution, then my advice is to use it first and use the breeding for the difficult tasks. And it's really important to understand the environments that your crops are experiencing, because it provides a great deal of insight about where you might want to go in the future. Finally, I'd like to acknowledge the huge number of people who've contributed to this work and indicate some acknowledgement of funding, both from the University of Queensland and the Queensland Government through the Coffee Initiative, the Australian Government through the Australian Research Council and ACR, the Grains Research and Development Corporation, which involves input from the federal government and farmers, and the Bill and Melinda Gates Foundation. And with that, thank you, and I hope you enjoy the rest of your conference.
Muchas gracias, David, por la participación en este simposio y por la muy interesante charla describiendo los sistemas de producción australianos y también cierta analogía con lo que pasa en Argentina.